Hi there. Thanks for having me. I'm so excited to be here. My name is Jagdeep Sidhu, and I'm an assistant professor at South Dakota State University. And I'm so excited to be presenting at this fifth annual Congress on Plant Science and Biosecurity. And special thanks to Mr. Peter Griffin for inviting me to this wonderful conference. And today I will be talking about cortical cell size and how that regulates root metabolic cost. Just to put things in context and give you an outline, I'll talk a little bit on the importance of root metabolic cost, especially in terms of soil exploration. And then I'll head into cell size, where I will present two stories, one on more on the mechanistic side of cell size and how that regulates root metabolic cost. And the second one, how cell size plays an important role in wheat polyploids, and especially during the wheat domestication process. And then as an outlook, I will talk about some of the related root anatomical phenotypes to cell size, which also regulate root metabolic cost. So just to put things in context, what my lab focuses on and what I have worked on for over a decade now are abiotic stresses. And one of the major being drought. As we know, it has an devastating effect on crop production, and it's only predicted to get worse as we head into the next decades of this 21st century, as shown here in red and purple on the maps of the next decades. And then on top of that, we have a problem of soil degradation, especially in areas which are heavily cultivated. And you can see here we have US Midwest, heavily cultivated area, all lined up in very degraded soils. And that creates a challenge for nutrient acquisition, especially for major nutrients, including nitrogen and phosphorus. Now, to understand how do plants can uptake these resources and how can we improve that, here is an distribution that we see in most of the agroecosystems, especially in the low input agroecosystems. So what we have is in the topsoil, we have phosphorus, immobile resources, and leach down into the deeper soil strata are water and nitrogen, and mostly in the nitrate form. And we have a traditional root system where it might be able to get one versus the other, but not both. You have a shallow roots can get the phosphorus, but deep they cannot get the deeper sources, vice versa for the deeper roots. And one intuitive answer to this might be that we can just increase, we can make both shallow and deep roots. But there's a problem with that, as these roots, both laterals and nodals, and all sorts of tissues are really expensive to both construct and maintain. And just to give you numbers on that, here is a below ground carbon budget of a genetic root system, where we have exudation taking up about 23% of the daily photosynthesis. And then you have growth taking up about 35%. And then a lot of that carbon goes towards root respiration and the respiration for maintenance of the tissue. And especially in abiotic stresses that I have just described, this can ramp up to 50% of the daily photosynthesis. And where is this respiration coming from? Let's take a closer look at a maize root system here. So what I'm showing is these are the nodal roots coming out of these stem nodes. And here's a longitudinal section labeled with major tissue types, and here is a cross section. Let's focus on the cross section here. And what we see is most of the root cross section is occupied by cortex. And those cells are expensive to both construct and then maintain. And there has been number of root cortical phenotypes that have been discovered in this last decade, which regulate root metabolic cost. And then certain phenotypes of those traits, they reduce root metabolic cost, and when used under abiotic stresses, they improve plant performance. And one such phenotype is root cortical cell size. So what you see on left here is you have maize under drought, and then on right you have a wheat under compaction, two independent studies. So you have large cortical cell side genotype, and then you have a small cortical cell side genotype here. And under drought stress, in case of maize, what you see is there's a reduction in root respiration as the cell size increases here on x-axis. And same is true for wheat under compaction, where under these lines represent different soil compaction stress. And what you see is there's a decrease in energy cost as the cell size increases. And this is really exciting because in both cases, not only decreased the root metabolic cost, but it also improved plant performance under drought for maize and under compaction for wheat. Now, when I started working with cortical cell size, what I was really interested in is how does cortical cell size regulate root metabolic cost? What's the underlying mechanism? And to understand that mechanism, we first developed this model called root slice. It's a functional structural model where we can manipulate root subcellular phenotypes as well as anatomical phenotypes. And what you see on the left here, you're seeing cell walls in this red, and this orange tissue is the uh, those little vacuoles. So we can go up to the vacuole and change the size of the vacuoles, size of the width of the cell walls, and many other tissues. So for this project, what we did, we simulated different cell sizes. So on left here, you have a root with smaller cell sizes. On the right, you have a root with larger cell sizes. And then we asked root slice, what would happen to root construction cost and root maintenance cost? And the first thing that was interesting to us was that the root slice predicted with increase in cortical cell diameter, there is a reduction in cytoplasmic volume and a corresponding increase in vacuolar volume. Now, as we know, vacuole is much more cheaper to maintain. It's compared to the active cytoplasm, where all the machinery is for, replication, your ribosomes and mitochondria and many more things. And root slice also predicted with that in decrease in cytoplasmic volume, there is corresponding decrease in mitochondrial density per root volume or cortex volume in this case, reduction in respiration, reduction in nitrogen needed to maintain and construct that tissue, and then a reduction in phosphorus. And these are function of cytoplasmic volume, so that's why they look all related. And that was one side of the story. We're also interested in what would happen if this is diameter, what would happen if we change the cell length? So we simulated different cortical cell lengths. So here's a model with shorter cells, and here's a model with longer cells. And here's an inset of those specific cells. And what we see is with the increase in cortical cell length, there is a reduction in cytoplasmic volume or cytosol, and a corresponding increase in vacuole. And the reduction in the cytoplasm leads to a reduction in the mitochondrial density, respiration, nitrogen, and phosphorus content. However, point to be noted that these percentage decrease in cytoplasm and increase in vacuole content is much smaller compared to what we have for cortical cell diameter. And that's because of the geometry. With length, you're increasing linearly. There, you are increasing with the power of 2. And with the modeling results, we confirmed our previous hypothesis, which were proposed in 2013 by Jonathan Lynch, that the larger cells have a lot of the volume occupied by vacuoles, which leads to reduction in the active volume. So here you have 3 smaller cells, and what could have been occupied by vacuole is now being occupied by cytoplasm. And this is not new as such, this scaling mechanism. It's shown in many other species. So you have yeast here, where the cell volume and the cell surface area scales pretty well with vacuole volume and vacuole surface area. And now we wanted to test this in using our maze lines, whether this phenomena occurs, whether this scaling occurs there as well. So We do see variation for cortical cell diameter, cortical cell length in wheat and maize. Here I'm showing two panels of maize. One is a maize IBM rill in yellow, and then a pink a maize vivid panel, which is much more diverse, and a wheat Watkin collection, which is again a diverse set of land races. And in all of them, we see a significant variation for both cortical cell diameter and cortical cell length. And in terms of relationship, we didn't see any relationship between the diverse panels, Medvedev and Watkin collection, but there is a slight negative relationship between cortical cell diameter and cortical cell length, which we need to explore further in terms of what that means. But out of this, we can select lines, especially from the Maze IBM rail, which differ for cortical cell diameter and cortical cell length. We use those lines, we call them isophenic lines here, uh, the lines which only differ for cell diameter, those are on top, and on bottom, they only differ for cell length and not for cell diameter. And using those lines, we did empirical measurements for root respiration, where the lines with in with larger cell diameter, they have reduced root respiration, reduced root nitrogen content, and lines which are longer in terms of cortical cell length, there is decreased root respiration and decreased nitrogen content. So it sort of proved the hypothesis that was coming out of um, the other studies and as well as the model, root slice model. And then we did some SEM imaging on these lines, which differ for cortical cell size. So what you can see here is a small uh, cortical cell, and you have a large cortical cell. And this distance between the cytoplasmic width, I would say, that stayed constant, which means that with increase in cellular cross-sectional area, there is a drastic increase in vacuole volume or vacuole surface area in this case, but not so much increase in the cytosol, which again goes with the hypothesis that with increase in cell size, there is a huge increase in the vacuole, which is cheaper to maintain, but cytoplasm is not so much. So that was first half of the story where we used a model to predict that the vacuoles are involved in reducing root metabolic cost in case of larger cell size genotypes. And then I'm going to talk about the another story here right now, which is how ploidy reduces root metabolic cost. Now that's counterintuitive. That's why I posed it as a question. The why ploidy doesn't lead to increase in demand for tissue construction and maintenance. And why this question? Let me explain. So nucleic acids are a large pool of phosphorus. Given here, about 40% of a cellular phosphorus goes towards nucleic acid. Now, as you can imagine, with increase in ploidy, there is increase in nucleic acid content. So you might expect that there is increase in needed phosphorus to maintain or construct that cell. And so it's true for nitrogen. In this case, nitrogen, most of the pool goes towards protein content. And we have seen from other studies that with increase in genome size, there is increase in log leaf N content, though this relationship might vary depending on species, especially in this paper. But there is a positive relationship between genome size and nitrogen content. So with that, what you can assume is diploids might need less phosphorus, less nitrogen compared to polyploids in order to construct their cells and maintain the cells. But that's kind of opposite to what we see in nature, or at least in the domesticated crop species. We have a lot of polyploids. So how are they maintaining their success? And many of them evolved in low fertility areas. And to test that theory, we used domestication series of wheat where we have diploids and then giving rise to tetraploids and tetraploids getting crossed with another diploid giving rise to hexaploids. And in each of those whether being diploid, tetraploid, or hexaploid, we have examples of wild as well as cultivated. So it's really interesting series for testing some of the questions that we were asking. And what we looked at is seminal roots, because easy to sample, and we can do a lot of genotypes with that. And contrary to what we believed or hypothesized, its increase in ploidy led to decrease in phosphorus content, nitrogen content, and root respiration. And when we took a closer look at how does the root anatomy look like, not surprisingly, already reported in literature, there is increase in cortical cell size as we increase as the ploidy increases. And what we see is with that increase in cortical cell size or cortical cell diameter, there is a reduction in phosphorus content, nitrogen content, and root respiration. So increase in cortical cell size leads to reduced nitrogen, phosphorus, and respiration. Similar story what we saw in maize, where they were not due to ploidy, but inherent increases in cell size. In this case, these cell size increases are induced by ploidy. So to further confirm that, we used root slice, where we simulated the actual cell size phenotypes for each ploidy level. And with actual phenotype, what you see is there is a reduction in nitrogen content and phosphorus content with increase in ploidy level. But when we switch the cell size of diploids and tetraploids to match the cell size of a hexaploid, that effect goes away, which clearly shows that the cell size is the main factor that explains the reduction in phosphorus and nitrogen content. And then to see whether it is due to ploidy or due to some other domestication events that have happened over the 10,000 years ago, we use the synthetic hexaploids and synthetic tetraploids, which the only difference from their parents is that they are polyploids and no domestication or other events have happened on them. And what we see for tetraploid, there is increase in cell size uh, against both the diploid parents, which corresponds to reduction in phosphorus content of roots. But for hexaploid synthetics, there is a significant increase in cell size compared to the diploid parent, which correspond to reduction in phosphorus content. But when comparing a tetraploid parent to the synthetic, there's no such increase or not a significant increase in cell size, and which correspond to not a significant increase in phosphorus content. Now, when we grow these natural polyploids and the diploids under suboptimal phosphorus and nitrogen availability, what we see there's a reduction in sensitivity for polyploids. So here shown as shoot biomass reduction compared to the control conditions. Under suboptimal P in black and suboptimal N in red, we see that the polyploids have a less shoot biomass reduction under these stresses compared to the diploids. And just to summarize, what we have is larger cortical cell size, utility under drought, compaction, and low fertility, because it reduces the energy needed to explore soils. And then we have natural variation for both cortical cell diameter and length. And we have seen maize and wheat, but we predict in other species as well. Vacuolar occupancy is the main mechanism by which cell size leads to reduction in root metabolic cost. And then ploidy-induced cell size increase in wheat is a part of a domestication syndrome, which I didn't really explain in this talk, but will be coming out as a preprint soon. And now as an outlook, I wanted to mention some of the other things my lab is working on, where we try to link multiple scales, starting with the gene to subcellular to anatomical phenotype. You saw that example for linking vacuole to the cell size, and then architecture being root depth, angle, and other things, and function being heat stress, drought stress, although fertility. And we specifically focus on anatomical and subcellular phenotype, and there we focus their usefulness under drought, carbon capture, low fertility, soil health, and because these are really underexplored, so we want to do them justice now, and want to discover these phenotypes and their utility to increase nitrogen and water. And there we focus their usefulness under. I'll take any questions.